Movement of substances into and out of the cell. The membrane's regulation of what enters and exits the cell is referred to as selective permeability. There's a constant movement of molecules across the membrane. Nutrients must move into the cell while wastes move out. However, very few molecules can pass freely across the bilayer. In order to do so, a molecule must be small and lipid soluble. Important examples for this class are oxygen and carbon dioxide. Molecules that are polar, charged, or large cannot pass freely, or at least not very quickly, and require the aid of a transport protein. Transport proteins can be either channel proteins or carrier proteins. Channel proteins form a tunnel through the membrane through which hydrophilic substances can pass. Examples of channel proteins include ion channels and aquaporins, which are special pores to allow water to pass through the membrane. Although water is a small molecule, it is polar and cannot pass through the hydrophobic core of the bilayer fast enough to meet the cell's needs. Carrier proteins bind to a specific molecule and shuttle it across the membrane by changing shape. An example of a molecule that crosses the membrane with a carrier protein is glucose. Glucose enters a cell 50,000 times faster with a carrier protein than it can without it. Mutations in transport proteins are linked with a number of different disorders. For example, cystic fibrosis results from mutations in a chloride transporter called the CFTR channel. Mutations in glucose transporters may be linked to a number of disorders, while defective aquaporins are associated with kidney dysfunction. As you probably know, our kidneys are the organs that regulate urine output, so the ability of their cells to take up or expel water is of great importance. Transport proteins are extremely specific to a particular substance. Glucose transporters, for example, are so specific that they reject even fructose, which is a structural isomer of glucose. The mechanisms behind the specificity of transport proteins is not fully understood in many cases and is the subject of ongoing research. However, I like to share an example I learned about in grad school because at the time I thought it was one of the coolest things I had ever learned, and that is how a potassium channel can be specific to potassium ions. Potassium channels will let only potassium ions through, but not sodium, even though both are positively charged ions and sodium is smaller than potassium. So you would think sodium would be able to sneak through a channel large enough for a potassium ion, yet it doesn't. Remember that in living systems, reactions are taking place in an aqueous environment, so ions are not actually floating around by themselves, rather they're surrounded by water molecules in what we call a hydration shell. A potassium channel is just the right size for a single potassium ion to fit through, but in order to do so, it must temporarily shed its hydration shell. The lining of the channel consists of oxygen atoms that precisely mimic potassium's hydration shell, so it can temporarily shed its water molecules and pass through the pore without resistance. Sodium, on the other hand, is not a perfect fit and will not give up its water molecules. Similarly, studies on the structure of an aquaporin, a channel protein specific to water, show that water molecules must pass through a portion of the aquaporin single file by turning sideways. The presence of the positively charged amino acid arginine prevents positively charged ions from sneaking through the aquaporin, while at the same time attracting the partially negative oxygen atom on the water molecule. Now that we're familiar with examples of transport proteins, let's look at the different ways substances can cross the cell membrane. Substances can move across the membrane either through passive or active transport. Passive transport is the diffusion of a substance across a membrane without any input of energy from the cell. It relies on the presence of a concentration gradient. Active transport, on the other hand, is necessary when a substance needs to be moved against its concentration gradient. This requires energy expenditure by the cell. You're probably familiar with the concept of a concentration gradient, which is a difference in the density of a substance in two adjacent areas. Substance is naturally diffused from areas of high concentration to areas of low concentration, or what we call going down their concentration gradient. It's important to note that each substance moves down its own concentration gradient and is unaffected by the presence of other substances. So think of it this way: if I cook broccoli in my kitchen, I like broccoli, but I hate the smell. It stinks up the whole house because the stinky molecules diffuse out of the kitchen and into other areas of the house. Meanwhile, odors coming from my cat box diffuse out of the cat box room and into the kitchen. So basically the whole house ends up stinking. If you have any questions about any of this content, please don't hesitate to contact me.